Good morning, Year 9. We're going to be looking today at the factors that are affecting enzyme function. Now, what I would like you to do is you're going to watch the video, and when we come to a piece of work, I would like you to pause the video and have a go at the work. If you want to do the work on the Google Doc, then all of the worksheets are saved onto the Google Doc. You can save it to the Google document and then submit it at the end of the lesson. If you want to do the work by hand, you can pause the video and then print off the sheets, do them by hand, stick them into your book, take a photo of it and upload it to the Google Classroom. So the first thing that we are going to start with, we've got a knowledge recall quiz. Now you know how these knowledge recall quizzes work. You've got 6 questions that I want you to answer. So I'd like you to pause the video now and have a go at the questions. So you should have had a go at the questions now. You've paused the video, you've had a go, and we're back in and we're going to have a look at the answers. So the first question said, define what a catalyst is. And a catalyst is a substance that speeds up chemical reactions but doesn't get used up in the process. The second question said, what is the chemical or the reagent that is used with starch? And the answer to that is to test for starch. The answer is iodine. The third one, what's the name of the tube that's going from your mouth down to your stomach? That's your esophagus. The fourth question, define active transport. So we've got the movement of particles going from, you might have said, a low concentration to a high concentration. So because it's going up a concentration gradient, that does require that it uses energy. Fifth question, what's the function of a nerve cell or a neuron? It is to carry electrical impulses. And then the sixth question, what's the function of a cell wall? It strengthens the cell and provides support. Moving on, we'll do a little bit of recap work now. So you've learnt last week in your Google lesson and also in class, we've talked about the lock and key theory of enzyme actions. So I'm just going to do a quick diagram and you should be able to do your own diagrams like this as well. So we've got the enzyme and I'm going to make my enzyme, it's just going to be that shape. So here's my enzyme. I'm going to label that enzyme. And then we've got a substrate, go for a different colour, that needs to fit inside that gap. So I'm going to label that the substrate. So this substrate fits inside this enzyme. It's not an exact match on my picture, but you get the idea. And the bit that I'm going to highlight now, this bit is known as the active site of the enzyme. So that bit is the active site of the enzyme. So that's stuff that you already know. We know about the lock and key theory. So we're going to have a quick go over today the effect of temperature on enzymes. So if we've got an enzyme and we heat it up, what effect would the temperature have on it? So I'm going to sketch a quick graph. Okay. And I'm going to have increasing temperature along the bottom. And then up the side, just the rate. So how quick the reaction takes place. So they've got the rate increasing and we've got temperature increasing. And we get this sort of pattern. As the temperature increases, the rate increases. And you can have a think to yourself, why is that happening? And think back to what you know about particles and collision theory. The particles are moving more, so they're more likely to bump into each other more. As it gets hotter, the particles gain more energy, they move more, they bump into each other more, they're more likely to react. It gets even hotter, they've got even more energy, they're more likely to bump into each other. Until with temperature, with an enzyme, we reach a point where it really quickly drops off. So this temperature here, we could call that the optimum temperature. That's the best temperature for this enzyme to take place. And then after that temperature, something happens that causes the enzymes to stop working. So they've been getting faster and faster. The rate's increasing as the temperature increases until it reaches this point. And what we would say, after this optimum temperature, the enzyme has been denatured. And you should know that when an enzyme's been denatured, that means that its active site, so this shape here, its active site has changed, so the substrate no longer fits inside the enzyme. So we've got a quick sketch there of what happens to temperature, the rate when we increase the temperature. Now, a couple of other things can affect enzyme action. The pH can have a similar effect to temperature. Different enzymes work best at different pHs, and also the concentration. The more you've got of an enzyme, the quicker it's going to react. The less you've got, the less it's going to react. And that kind of makes sense. So we've got three things that can affect enzyme action: temperature, pH, and concentration. Now, what I would like you to do now, and there's a copy of this saved on the system and also inside the Google Doc, so it's saved inside that Google Classroom or in the Google Doc, depending on if you want to type or to handwrite. I want you to spend 10 minutes having a go at this question. So the question's about factors that affect enzyme action. So you to pause the video and spend 10 minutes having a go. So I'm going to assume that you have spent 10 minutes having a go, and we're going to have a look at the answers now. So an enzyme is added to a solution of the substrate. What factors affect the rate of reaction? And it says tick as many of the boxes as you need. So what could affect in it? Well, we know that the temperature is going to have an effect on it. You also know that the concentration is going to have an effect on it. And then the last thing that's going to have an effect on it, we know that the pH of the solution is going to have an effect. So then if we move down and we can have a look, you should be green penning your work as you're going along. We'll have a look at part B. So the graph shows an enzyme controlled reaction, just like the one that I've just drawn in my book. Circle the correct bold temperatures in the paragraph to explain what it shows. So between 0, 10, 37 and 54, the rate of reaction increases. So it's increasing, it's going up between 0 and between about, so when's it increasing? That's about there, so about 37. So we want 0 and 37, the rate of reaction increases with the increase in temperature. And you can see that we've got a roughly straight line going up there. What is the optimum temperature? Well, you can see there, the optimum temperature, just like on my chart, was 37. And then we've got that last question then, higher than 10/37 degrees, the rate of reaction decreases with the increase in temperature. So once we get past 37, that rate of reaction does decrease. And then the last in, at around what temperature does the enzyme stop working? Well, you can see there, well, what do we think? Is it going to be, it has completely, there's nothing going on there at all. Is there any rate of reaction there? Well, there is a little bit going on there. So at around 54 degrees, the enzyme stops working. So there's no activity going on there at all. Why do enzymes stop working over a certain temperature? Does it die? No. Does it melt? No. Does the substrate evaporate? No. The active site changes shape. And what do you need to remember? So we've got some missing words to fill in here. Enzyme activity is affected by something and pH, so we're going to have temperature in there. The temperature that an enzyme works fastest at is called its, that's going to be its optimum temperature. High temperatures and changes in pH, something the enzyme. What could we say there? High temperatures are going to denature the enzyme, changing the shape of the active site. This means that the substrate can no longer bind to the enzymes, and then you've got the last sentence there. So we've done a couple of the tasks for today's lessons so far. Okay, so next activity for us to have a look at, we've got an exam question. This is saved in the Google Docs, and also you've got a separate copy of it if you want to just print it out. So we've got 3 different enzymes: enzyme X, enzyme Y, and enzyme Z. We've got pH this time along the bottom, so I'm going to highlight that. And then it's got activity, so activity is the same as rate of the enzyme. And it wants to know which is the best pH for enzyme Z. Now, I don't think I've got a ruler handy with me, so I'm just going to use my pencil, and I'm going to do that and scroll down. And if I followed that through, it's about there. Followed that through to 8.2, 8.4, 8.6. So the best pH for enzyme Z is 8. Oh, you can't see my answer, 8.6. The stomach makes a substance that gives the correct pH for enzyme action. Name this substance. So you could just put acid. It would be better to put hydrochloric acid, because that is the name of the substance that is produced in there. The question then says, which enzyme will work best in the stomach? So let's have a look. Acids are at a low pH, so we want something that works less at a low pH. Which one works best? It's going to be enzyme X. Works best at a low pH. And then question 4 requires you to think a little bit for question 4. It says, describe how different parts of the digestive system help to break down molecules of fat so that they can be absorbed into the body. So we need to be having a think here about these different things. So your answer, you should have been writing about the enzyme is lipase, and lipase is made in the pancreas and the small intestine. You needed to have said that lipids or fats are broken down into fatty acids and glycerol. And you should have made mention as well. You should have also talked about bile. Now, bile is a substance that's made in the liver, and it's stored in the gallbladder. And if you remember, bile does a couple of things. One thing that bile does is it breaks up large fat droplets into small fat droplets. That's one of the jobs that bile does. The second thing that bile does is it increases the pH, so that when the enzymes when it's come from the stomach into the small intestine, the enzymes that are in the small intestine want it to be at a much higher pH. Lipase wants it to be at a higher pH, and what the bile does is it increases the pH. You could have said that the products where this takes place, so the digestion is in the small intestine, so all fat digestion takes place in the small intestine, and that's probably the things that you would have wanted to have said. So we've talked about, you'd have talked about the enzymes, you'd have talked about bile, you'd have said where digestion takes place, where the enzymes are produced, the names of the enzymes, and if you've included all of those things, then you'd have got a really nice answer that covers all those different parts. Now, there's one last thing that you can have a go at. This is more of an extension activity, and you will need to kind of, let me get it so you can actually see it. Pause. I'm struggling with the light here. pause the video and have a go at these questions. These are not in the Google Doc, so if you've got this far and you want to have a go, then pause the video and have a go at these questions. And then when you've unpaused it, if you want to green pen your answers, here are the answers to those questions. Now what you need to do with all of today's work is submit today's work so that I can see how well you've done, and make sure that you have got your green pen answer work on there so that I know that you've watched the video. Thanks a lot, Year 9, and I'll see you again.